Hey everyone, so we are picking up where we left off on the nervous system PowerPoint, and I've actually made some edits to this slide because it wasn't super clear, especially about fast axonal transport. So fast transport can be anterograde or retrograde, but of course anterograde can't be in either direction, so I fixed that. And we are going to pick up right where we left off. So starting with axonal transport again, just recall that the proteins that a neuron makes are made in the cell body because that's where the nucleus is. But they have to be transported at a huge distance down the axon to the axon terminal because the axon terminal is sort of the business or sending end of the neuron. So that can be really, really long in some nerves. Consider one that goes from your sacral plexus all the way down to your foot, for example. So the axonal transport system is basically in place to carry things down the axon or back up from it. So fast axonal transport is exactly what it sounds like. It's fast. So let me get my pen down here. So it is fast, and of course fast here is relative. So 400 millimeters a day, and by here they mean 24 hours. Isn't that fast compared with regular human life stuff, but in the context of a cell and movement of things by motor proteins, it's pretty darn fast. So organelles can travel this route, so if there are more mitochondria that are needed in the axon terminus, they get there this way. Enzymes and also vesicles, typically these vesicles are full of neurotransmitter, and small molecules for other purposes. There is fast retrograde transport, so retrograde means toward the soma. And this is for recycled materials and pathogens. So I mentioned in lecture and in the last PowerPoint slide, sometimes these pathogens include viruses, especially ones like herpes that like to hide in the dorsal root ganglion. Slow axonal transport is also called axoplasmic flow. So the axoplasm is the cytoplasm in the axon. And this moves cytoskeletal elements and new axoplasm down at about 10 millimeters per day during repair and regeneration. So here's a picture of axonal transport. So in this direction, we're going away from the nucleus toward the terminus. So this is going to be anterograde. And notice we have motor molecules, kinesin and dynein, transporting this vesicle down the microtubule. And in this case, the vesicle is full of neurotransmitter. So that's probably going to be exocytosed down here. When the neurotransmitter is taken up again, that's called reuptake. The neurotransmitter is inside this vesicle. If this is worn out neurotransmitter, it needs to get disposed of. And in order to maintain the appropriate amount of membrane, because every time you do exocytosis, you're going to add a little bit of membrane to this end, you have to subtract membrane at the same rate as you add it to keep the membrane amount the same. So that's part of retrograde transport's function and purpose. So this slide is concerned with naming things according to location. So the entire nervous system, both the central nervous system and the peripheral nervous system, contain the same kinds of cells. So we've got glial cells and neurons, but they have different structural shapes depending on whether you are in the CNS or the PNS. And accordingly, we name those structures differently. And this is what we're going to cover now. So a tract is a bundle of nerve fibers within the CNS. So these are not including perineurium or epineurium or any of the connective tissue coverings. They just run together in a group. So individual nerve fibers potentially extend through a nerve and out of a tract or pass into the CNS this way. So for example, something might be called a tract as long as it's confined to the spinal cord, but once it leaves the spinal cord, it becomes a nerve, and vice versa. So white matter is myelinated. That's because myelin is made of phospholipids, which are fats. And if you've ever looked at raw bacon, the fat part looks white, doesn't it? So that's why it's called white matter. So in the central nervous system, we call white matter myelinated tracts. In the peripheral nervous system, we call white matter myelinated nerves. But the term white matter itself is typically reserved for central nervous system structures, so the brain and the spinal cord. Gray matter takes two forms, and this is unmyelinated nerve fibers such as dendrites and some axons, as well as cell bodies. So in the central nervous system, we call these nuclei. So it typically looks like an island of gray matter surrounded by white. In the peripheral nervous system, we call these ganglia. So a ganglion is a swelling along a nerve that indicates the presence of cell bodies within. So a great example of this is the dorsal root ganglion. Dorsal root ganglions. This is a swelling of cell bodies. These are the cell bodies of unipolar sensory neurons, and the cell bodies of those neurons live right outside the spinal cord in the dorsal root ganglion. All right, so nerves and tract function style. So we just talked about structural differences, but now we need to talk about functional differences. So this is where that SAME-DAVE acronym comes in. So let me remind you about SAME-DAVE. So SAME stands for sensory equals afferent. So that's carrying things toward the CNS. And motor neurons, that's what the M is for, are efferent. So within an individual neuron, the direction of the impulse is confined, and it's determined by the relative location of the cell body and the axon terminals. So you can't have a sensory nerve turn into a motor neuron or vice versa. But if it's in a nerve, so many axons in a bundle, or a tract even, those can be mixed, meaning it's one large structure containing lots of nerve fibers running in parallel to each other, and some of those nerve fibers are carrying impulses toward the CNS and others are carrying impulses away from the CNS. So most spinal nerves, for example, are mixed, and a handful of cranial nerves as well. Glial cells are also called neuroglia, or just glia for short. I tend to just refer to them as glia. It's most parsimonious. So these are supernumerous, and they make up half the volume of the central nervous system. So that's considerable considering how neuron dense the central nervous system is. These are much smaller than neurons. They are 50 times more numerous, which is a lot considering how many neurons you have. And the cells can divide. So adult neurogenesis, so the growth of new neurons in grown-ups, does happen. It just happens very rarely and very slowly compared to the rate of mitosis in other body tissues. So for a long time, scientists just said, well, they don't ever divide. Once you have all your neurons, you keep them or you lose them, but you don't get new ones. That's not actually true, but they still do so much more slowly than glial cells. So it's still a distinction between the two that you can make. In tumor formation, which we call gliomas, so this is a glial cell tumor specifically, so it's glial cells that have gone rogue. In glial cell tumors, those glial cells are undergoing rapid and uncontrolled mitosis, so they're turning into a mass and they're not doing their jobs. There are 4 cell types of glia in the central nervous system, and those are astrocytes, oligodendrocytes, microglia, and ependymal cells. There are only 2 cell types in the peripheral nervous system, and those are Schwann cells and satellite cells. So if I ask you to compare and contrast the glial cells of the CNS with the glial cells of the PNS, one contrast you might make is that there is higher cell diversity, 4 kinds, in the CNS versus only 2 in the PNS. So we're going to go through each individual one of these and tell you what they do. Astrocytes are the most numerous. So these are by far outnumbering both neurons and the other glial cell types. These help to form the blood-brain barrier by covering blood capillaries with their little cell feet. So pseudopods, which pseudopod means pretend foot. So these guys reach out their little cell feet and they wrap around blood capillaries to provide an additional layer so that what comes out of the blood in the blood capillaries in the brain is very tightly controlled. And that helps prevent pathogens and toxins from having access to and damaging your brain tissue. These can also metabolize neurotransmitters. So if you're constantly neurotransmitting, which your brain always is, having cells to help with that is important. They regulate potassium balance in the extracellular fluid, and this is very important because potassium has the greatest influence on membrane potential. It's a terrible end. There we go. I'm going to abbreviate this RMP. They provide structural support as well, so they can brace neurons away from each other, and they transfer nutrients to the neuron from the blood. So it's another layer of protection. Because nutrients are really greedy for glucose and nutrients, but the delivery system, the blood, is a potential source of hazard for the tissue. So you kind of split the difference by having astrocytes be the gateway. All right, oligodendrocytes. Now it says most common glial cell type, and one of the students in my 231 class rightly was confused because most numerous sounds a lot like most common. This means that they're the kind that you can actually see in a microscope slide the most frequently, so most of the time you don't see astrocytes, but glial cells that do pop up in nervous tissue cross-sections. So this is a cross-section of brain tissue. And this would particularly be in the white matter because that's where myelin is. So common means most commonly visible under a microscope, not most numerous. So each of these forms a myelin sheath around more than one axon, and that's one of the major differences between them and Schwann cells. Schwann cells also myelinate cells, but they only do so in the peripheral nervous system, not in the central nervous system. And we'll look at a picture that compares them a little bit later as well. So the reason that they're so commonly seen is because an individual cell can reach out and myelinate a couple different axons. So maybe this oligodendrocyte's foot is myelinating this axon, but also this one and this one and this one and this one. So all the little white spaces here are part of the same cell, perhaps. Microglia are what they sound like. They're very tiny, and they're found near blood vessels. So if you look at this picture, you can see some blood vessels, and you can also see some microglia. So they tend to look similar to the parietal cells of the stomach in that they sort of resemble a fried egg with bright pink cytoplasm. So these are phagocytes, and they specifically remove dead cells or debris from between and among neurons, because if debris builds up between and among neurons, synaptic transmission is impaired, and that causes problems with cognition and motor function, among other things. So you want to get rid of that gunk. These are actually derived from cells that give rise to macrophages and monocytes, so that cell is called a monoblast. So unlike the other glial cell lines, these ones come from the same cell family as some white blood cells. And that's because most cells that are avidly phagocytic come from some phagocytic cell line. So there's kind of cell dynasties from the perspective of stem cells and their ancestors and progenitors. Okay, ependymal cells. You may also see this pronounced ependymal cells. That's kind of a style choice. I don't really care how you say it as long as you can spell it. So these are actually, if you notice, simple columnar epithelium. And it is ciliated. So if you're wondering why I made you learn all the epithelial types in the beginning, why would you ever use that? Well, epithelium is all over the place. It's super ubiquitous. You're going to have to know it and continue to know it going forward. So here you can see those little cilia. This is a bit of the spinal cord. So here we have the central canal, which is the hole in the middle of spinal cord. And it contains cerebrospinal fluid. So these guys help to produce and modify the cerebrospinal fluid. They participate in CSF production along with another structure that we'll learn about later called the choroid plexus. So they manage all of the fluid in the cerebral cavities and central canal. And so your cerebral cavities are large spaces in your brain that are full of cerebrospinal fluid. So this is what they look like. Now we are moving into PNS glial cells. So peripheral nervous system glial cells, and I'll just write that up here. There are only two kinds. And these guys are flat cells that surround neuronal cell bodies in peripheral ganglia. So what does peripheral ganglion mean? So peripheral means it's in the PNS. And ganglia are collections of, that's so terrible, N, cell bodies, specifically neural cell bodies. So neural cell bodies in the peripheral nervous system are just sitting in the tissue, unlike those in the central nervous system where there's this robust barrier system of meninges and of blood-brain barrier stuff. The peripheral nervous system cell bodies are relatively unprotected, so it helps them to have a little population of liner cells. So this is pointing out neuronal cell bodies, and these little cells, you can mostly only see the nuclei around them, are the satellite cells. So what that looks like is, let me draw a sensory neuron here. Sensory is its function, unipolar is its shape. Give it a nucleus. So the satellite cells are going to wrap around this neurosoma, kind of like petals of a flower, to keep it nice and protected. So that's what those look like. Schwann cells encircle peripheral nervous system axons, forming myelin. So let's go in and do that. So each cell produces part of the myelin sheath. So what does it mean, part of? Why isn't it the whole myelin sheath? It takes many, many Schwann cells to form a complete myelin sheath. So the myelin sheath formation begins during fetal development, but it proceeds most rapidly during infancy. So when a neonate is born, the nervous system isn't sort of fully cooked yet, and it continues to develop as that infant grows and thrives. The area of bare axon between adjacent Schwann cells is called the node of Ranvier, and Ranvier was the guy who found them. So what you're seeing in this picture is a cross-section. So this is a cross-section of a neuron. So this part is the axon. This is the nucleus of the Schwann cell, so let's underline that. And then this cell, the Schwann cell, is sitting on the axon, and it's wrapping its membrane around and around and around and around and around the axon. And here's the axoplasm. So you can actually see little vesicles, these little round things, and then this is a mitochondrion. So these are presumably being carried down or up the axon via axonal transport. This is what myelin looks like, so it's a jelly roll style wrapping around the axon. So, myelin sheath. It's an insulating layer around a nerve fiber, and its primary function is to speed up excitation travel. So it speeds up the rate at which action potentials can bounce down the axon. So let's put this. So I want to stress that this only has to do with action potentials. The myelin sheath has no impact or influence on the processes at the terminus in a chemical synapse. So no effect on exocytosis of vesicles or any of that. It just helps the action potential get to the end faster. So myelin sheath is formed by oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. And it looks white, as I mentioned, because it's fatty, and that's because it's 80% lipids, specifically phospholipids and cholesterol. And sphygmolipids, which is just another kind of lipid. So in the peripheral nervous system, hundreds of layers of a cell wrap the axon, and the outermost coil is the Schwann cell sort of body itself, which forms something called the neurolemma. It's also covered by a basement membrane and by endoneurium. So this is over the top of the Schwann cell, and we'll see this in neuron repair. In the central nervous system, there's no neurolemma or endoneurium, there's just oligodendrocytes, and that's because extra connective tissue going over the axon would just get in the way of neurons in your CNS talking to each other, so we just don't do that. The nodes of Ranvier are the gaps between myelin segments, so either between feet of oligodendrocytes or between entire Schwann cells. And the initial segment, which is the area before the first Schwann cell, and the axon hillock form the trigger zone where signals begin. So if we have a cell body, and I'm going to make it a multipolar neuron kind of awkwardly here. There we go. And then there's our axon hillock, and myelin starts here. And then we have a node, and then another myelin bit. So these are going to be Schwann cells. The initial segment and axon hillock is this bit right there. So here is what this looks like. So the neurolemma is the outermost layer of the Schwann cell, and the rest is the actual myelin sheath. So this is part of the same cell, but this part is much more tightly wrapped in a clump, whereas this allows the cell to have some cytoplasm available to transport things. So that's the neurolemma. Covering the surface of that is the endoneurium, which is connective tissue, and that is connected to the Schwann cell by the basal lamina. So this is, of course, a Schwann cell because no endoneurium and no basement membrane or neurolemma in oligodendrocytes, because the oligodendrocyte cell body is over here, away. It doesn't need to have a neurolemma to facilitate its function because it's not squishing itself. So here we have the myelination for this one, and then this one, and then this one. So this oligodendrocyte is myelinating multiple different axons. Okay, so moving right along, let's talk about nerve fiber repair. So unfortunately, this drawing is a little bit stretched out. Not sure why. It looks a little bit warped on my screen. So basically what this is showing you is a motor neuron. How can I tell it's a motor neuron? Well, it is multipolar, and it's innervating a skeletal muscle, very obviously, so that's how I know it's a motor neuron. Okay, so you can see here there's a myelin sheath, so Schwann cell, Schwann cell, Schwann cell, Schwann cell, and there is an endoneurium, which we know is connective tissue. So this is going to protect this axon and its little telodendra over here. So what happens if you injure it? You break the axon at some point. Well, as long as you haven't destroyed the cell body, so I'm going to put this asterisk over here, as long as soma is intact, you can repair this. So here's what happens. Local trauma, here's a break. Macrophages come in to eat up debris. So these are going to eat cell bits. And dead cells. Cool. So, fortunately, we still have the endoneurium. So downstream of the break, before the repair happens, you've got this empty endoneurium with deteriorating axon and deteriorating Schwann cells. So the living cells go away, as does the axon, but you can see that there are little growth fibers or processes here. So these ones are new. The empty tubing of the endoneurium is called the regeneration tube. And basically, via axoplasmic transport, or slow axoplasmic transport, the growth processes are going to anchor the growing axon, and the axon is going to extrude down the regeneration tube to try and reach the end. Also note that there is a concomitant atrophy of muscle fibers that comes along with lack of innervation. So if you have a muscle that is denervated, so no longer innervated, so if there's no innervation, you get a loss of tissue equals atrophy. As the growth processes reach the end, they remake the neuromuscular junction, and once you can start exciting this muscle tissue again, you get a reversal of atrophy, and you get hypertrophy again, and the muscle presumably goes back to normal. Now this takes a while, but it does work as long as you haven't ruined the soma. So, regeneration of peripheral nerves can occur if the soma and neurolemmal tube is intact. The stranded end of the axon, so the part that's not connected to the soma, though that degenerates, as does the myelin sheath. And the neurolemma tunnels from the area of damage to the effector, whatever that is. In the last example, it was a skeletal muscle. There can be others, of course, because we know effectors can be smooth muscles or glands, too. So the macrophages remove debris. The new Schwann cells grow, allowing path for the development of the regenerating axon. And the stub puts out little sprouts. The growth rate increases as sprouts reach the tunnel, and also the sprouts help to stabilize the growing end. And until nervous connections are restored, skeletal muscles will atrophy. So that's the whole process in overview. So now we need to start to talk about the purpose of neurons. So why have them? Well, we know that we have them to send electrical signals, culminating with chemical signals. So neuron doctrine states that nerve pathways aren't continuous, so there's not a wire that goes directly from your brain to the muscle controlling your big toe, but rather a series of separate cells that play a game of cellular telephone. So if I decide to wiggle my big toe, one neuron, whose cell body lives in my motor cortex on my frontal lobe, is going to excite a spinothalamic neuron, which is going to excite another neuron, so all the way down to my foot. So communication is based on the mechanisms for producing electrical currents and potentials. So let's define these things. Electrical potential is a difference in charged particles between different parts of the cell. So what do we mean by different parts? Well, let's get my pen here. So different parts can mean in versus out, or it can mean one part of the cytoplasm versus another. So axoplasm A to axoplasm B. Electrical current refers to the flow of charged particles from one point to another within the cell. So if charged particles are going from axoplasm bit A to axoplasm bit B, that's a current. So currents imply movement. And a potential is just a difference, not yet being acted upon. So neurons are electrically excitable due to the voltage difference across their membrane. And if you want to know more about how you set up and maintain a resting membrane potential, go watch the video I made on that very thing. And we're going to get more into it in this PowerPoint as well. So if you're the kind of person who learns better by looking at PowerPoint slides, this might be more up your alley, but both of those resources are available to you. So two types of electrical signals exist. One are action potentials. These are the ones that have to be set off by a threshold stimulus or above and have a predictable and repeated pattern. These are also the ones that can travel down the axon in long distances. Graded potentials are local membrane changes. So let's say a little patch of membrane over here on the soma has some sodium ions that get let through, but only in that little patch. Only that patch of membrane is going to depolarize, and then if there's no further stimulation, it will repolarize itself. So graded potentials are limited to one membrane patch. So this is all due to flow of ions through ion channels in the cell membrane. So let's look at some channels. So leakage channels are always open. And this seems counterintuitive because you might imagine, well, if I want to keep some things in and other things out, why would I have leakage channels? Well, consider the following. Here, if we have the sodium-potassium ATPase, and that's going to kick out 3 sodium and let in 2 potassium as long as it has ATP, it's going to go until there's no potassium in the cell at all, and no potassium outside the cell at all, lots of sodium, and no sodium inside the cell at all, but lots of potassium. So it'll go to completion for as long as it has fuel to do that. So that's going to result in an increase in membrane difference until a plateau. But that's not ideal, and it doesn't reflect the negative 70 millivolts that the membrane maintains itself at. So by having a very small number, not very many, of leakage channels that always lets ions trickle out, or in, so sodium leakage channels allow sodium to trickle into the cell at very slow rates, and potassium leakage channels allow potassium to trickle out. That explains the resting membrane potential. So it explains how you can maintain a negative 70 millivolt constant resting membrane potential. Because there are slightly more potassium channels than sodium channels, the membrane permeability to potassium is higher. And as a consequence of that, even though influx of sodium ions is how an action potential is produced, potassium in the extracellular fluid is the most important effector of membrane potential. So if you have more potassium in the extracellular fluid than normal, due probably to ion dysregulation or nutrition issues, that's going to make your membrane more difficult to depolarize. So having and managing potassium in the appropriate body water compartments is important. So gated channels come in different flavors, and these different flavors correspond to what thing it is that makes the channel open. So gated channels typically let ions through, although sometimes other stuff too, and they can be voltage gated, which means they only open at a particular voltage range. So if the voltage changes in a way that they like, they'll open up. Ligand gated, so ligand just means chemical, can be a neurotransmitter, hormone, or just a small molecule. Or an ion. Mechanically gated ones are ones that open when they are mechanically stimulated, so when they're squished or bent or stretched in some way. So most of the baroreceptors or pressure receptors in your arteries have mechanically gated channels that result in depolarization in response to stretch. Stereocilia in the hair cells of your ears start to fire off changes in potential when they are bent, and Pacinian corpuscles deep in the skin, these start to fire off potentials when they are smushed. So that's how they detect pressure. So membrane potentials, looks like the following. We've been talking about this in the context of excitable cells, so neurons and muscles, but all cells maintain a difference of concentration in ions across the membrane. Just not all of them do so to the extent that they are routinely and habitually depolarizing and repolarizing. So an excess of positively charged ions outside of the cell membrane with internal membrane deficient in positively charged ions creates this potential difference, and it's called potential because putting different things on different sides of a membrane represents potential energy. So it's called a potential due to potential energy. And again, if you want to know about potential energy versus kinetic energy, go watch the resting membrane potential video for some more information on that. So polarized means that there's a plus side and a minus side. So if there's more positively charged ions outside of the cell membrane, that's the plus side. And if there are fewer positively charged ions inside of the membrane, let me draw myself a membrane here, that's the minus side. So if you want to measure this, you can take some electrodes at different poles. One you can place outside the plasma membrane, and the other one you can string into the cell, and then these two will measure the difference in voltage between the outside and the inside. And so because the inside is the negative side, the potential is reported to be -70. So that's kind of how that works. So the resting membrane potential is -70. And also notice down here, in addition to having lots and lots of potassium, but less total positive charge than is out here, we've also got large anions that can't escape the cell. So these are things like phosphate from the cleavage of ATP and such. Those all have negative charges as well, but phosphate is PO form negative, so that's big. Can't get through here. So unequal distribution of electrolytes is what makes up this difference. So different ratios of sodium to potassium and different ratios in addition to large anions. So if these ions could, they would diffuse down their concentration gradients, so sodium would enter and potassium would leave. But the membrane of the neurons and electrically excitable cells is selectively permeable. So the membrane is able to control very tightly what is able to move in and what is able to move out. So if you keep your ion channels shut, you necessarily will prevent diffusion from occurring and maintain a difference between this side of the membrane and this side of the membrane. So the membrane is very permeable to potassium, as we mentioned. And this will leak out until the electrical gradient created attracts it back. So this is one of the things that explains the resting membrane potential, is that it's leaky to calcium. Cytoplasmic anions can escape due to charge. So here we have phosphate, and this is sulfate, and there are also organic acids and proteins. The membrane is much less permeable due to fewer sodium leak channels to sodium. And the sodium-potassium pump pumps out 3 sodium for every 2 potassium returned. So there's a net loss of 1 unit of positive charge per pump. So net loss of 1 plus per ATP. And these work continuously, which require a really high need for ATP. And that's what makes neurons so glucose and oxygen hungry. So they use up these things a lot because they're always powering this pump. So this is why you should eat in class, because neurons need glucose. Okay, so local depolarizations versus action potentials. Local depolarizations move. So don't move, so they stay in the same spot, whereas action potentials travel, so they travel down a neuron. So one difference between action potentials and local potentials is that they are limited to a particular patch of membrane. So local disturbances in membrane potential can be stimulated by chemicals, light, heat, or mechanical disturbances, so it depends on what that particular neuron is primed to respond to. And depolarization decreases the potential across the cell membrane due to opening of gated sodium channels. So these could be ligand gated in the case of chemicals, or they could be chemically gated or something else. So sodium rushes in down the concentration and electrical gradients, and it diffuses for a short distance inside the membrane, producing a change called a local potential. So local depolarizations actually come in a couple different kinds. There are local depolarizations, and they vary in magnitude, so the arrow here corresponds to stimulus intensity. This is supposed to be 1T. Dang it. So for a big stimulus, you get a taller, longer depolarization. For a little stimulus, you get a shorter, smaller depolarization. You can also have a stimulus that hyperpolarizes. So this is still a local potential, but it's in the opposite direction, so local hyperpolarization, meaning this neuron is being pushed away from threshold and it's less likely to fire. So, summarizing the differences once more, graded means the amplitude is proportional to the strength of the stimulus, which is generally small, so you have to add these up in order to get to threshold and create an action potential if you want. We'll talk more about that later. They're also decremental, so they get weaker the further they spread, and that's another reason why a single graded potential can't make an action potential. They are reversible, so once the gated channels close, the membrane potential goes right back to normal, and they're also reversible because of the potassium leak channels. And as I showed you, they can be either excitatory or inhibitory. So if they're excitatory, a stimulus triggers the opening of sodium channels, causing the membrane potential to move towards zero, so get more positive, depolarize. If they are inhibitory, the stimulus triggers the opening of potassium channels, increasing the membrane potential, so hyperpolarization. So potassium channels are going to let potassium that has been sequestered out, which makes the inside of the cell even more negative. So in the next slide, we will start talking about action potentials in particular, but as we've reached the 42-minute mark, I will save that for the next presentation. So thank you for your attention, and I'll see you in the next video.